Hello and welcome to Nikolai's genetics lessons, and today's question is: influenza virus enters host cells where, and you have to choose the correct answer. And the interesting thing that multiple correct answers are possible here, but only one would be the best answer. So I want to start with answer B, injection of the polar tube. So what this might mean, and the only thing that this might remind you would be bacteriophage that might inject its own genome in the bacteria. But actually there is no such term as injection of the polar tube at all. So this is completely wrong answer. Let's now concentrate on the rest three answers. And now let me talk about phagocytosis. And different types of phagocytosis exist. For example, our cells can get water, small molecules like a sugar, through the pinocytosis, and phagocytosis would be engulfing of the large proteins or bacteria by, say, such cells as protists. And imagine that here we have bacteria, and here is protist, and when this protist, which is unicellular, would sense a food, and the next stage, what is going to happen, this protist would change its form, and once again on the next stage would completely engulf this. Once again, it can be bacteria or just proteins, and would digest it. And through the process that we call exocytosis, then would get rid of the material that it doesn't need for its own function. So it would release this bacteria and would use its proteins, its nucleic acids, amino acids, in order for its own functioning. So this is example of the phagocytosis. And actually many cells in our organism use this process in order to protect us from also bacteria and viruses, and we have many such specialized cells in our blood, such as neutrophils, monocytes, macrophages, mast cells, and dendritic cells. But once again, this is a broad term, and as long as answer A also can be a correct answer, but let me show you the difference. And now imagine that we have here surface of the cell, and here is a double lipid layer, and here is our virus, influenza virus. And influenza virus has surface proteins here, two types of surface proteins, hemagglutinin and neuraminidase. And in order to attach to the lipid bilayer, this cell must have also receptors to which these proteins, surface proteins of the virus, would have high affinity, and such receptors would be sialic acid, and this is type of sugars. So this is also glucoproteins. And what is going to happen on the next stage, actually this cell would start to engulf this virus by making more and more such receptor mediated endocytosis, and next stage we would see formation of the vesicle, which would contain virus, and it's going to have double lipid layer of the cell. So this vesicle would be formed including this double lipid layer that surround the virus. And what's going to happen next? Also, our organism protects itself, and our cells protects by releasing such viruses. So we would have another vesicle which has very acidic environment, which normally would kill and release any proteins. But actually influenza virus have learned how to use this lysosome vesicle that also includes enzymes that break down proteins for its own benefit. And now let me draw another picture, a little bit picture of this virus. So first of all, this virus has protein code that we call matrix, and outside this matrix has also lipid bilayer that this virus acquired when this virus exits the cell. So it's not something produced by virus, but something that this virus steals when leaves the cell. And inside this matrix we can find fragmented genome of the influenza, which consists of 4, 5, 6, 7, 8 fragments, and this is a negative RNA strand virus. And also on the surface we can find two types of the surface proteins. One we call neuraminidase, or NA. Sometimes you can see N abbreviation, sometimes NA. And another protein would be hemagglutinin, or H or HA. And actually this is also would be antigens. Also influenza has a name that has H and N, and some number that which follows these letters. For example, influenza H1 N1, which we had few years ago in 2009 pandemic, and this is where type A influenza virus. Actually, as you see, 3 variants are possible, whether it can be A, B, and C. And the variant of the influenza virus that is A influenza virus we can find not only in humans but also in many animals. So that's why this is most virulent virus that cause most harm. And now what happens at the stage 3? So this is first, second, and third stage. Actually, as I said, this lipid bilayer of the virus would fuse with cell membrane by lipid layer of the vesicle, and as you remember, also outer layer of this virus would be destroyed, and this single-stranded RNA fragmented genome would be released into the cytosol of the cell, and then would be transported into the nucleus of the cell. And now viral genome will take over the nucleus and would replicate itself hundreds and thousands of times, and would proceed with its own cycle, infecting other cells. So now let's return to our three variants of the answers. As you see, answer D, phagocytosis, could be the correct answer, but I would prefer to use this answer if we would talk about specialized in phagocytosis cells, professional cells such as neutrophils, monocytes, macrophages, mast cells, or dendritic cells. And by the way, this is how HIV virus enters, taking over T cells. That is also belong to this professional phagocytosis cells. But because influenza virus use different types of cells to penetrate and replicate, we rather have to choose answer C, receptor-mediated endocytosis. But once again, just remember that in literature and in textbooks and scientific papers, you can see that receptor-mediated endocytosis and phagocytosis as a mode of entrance of the influenza virus can be used interchangeably. So variant C, receptor-mediated endocytosis, would be more appropriate. And variant A, fusion with cell membrane, also, as you see, describes what happens on the third stage. Not on the first stage, but on the third stage. But before, it also would be fused with another vesicle that we call phagosome, that contains lysosomes. And as a result of this process, double lipid layer of the virus would fuse with double lipid layer of the vesicle. So fusion with cell membrane. But once again, this is only description of what happens at the stage 3. So now we can choose the correct answer, and as you see, the answer most appropriate, or the best answer, would be receptor-mediated endocytosis. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.